Hello, everyone. Welcome to our last session. We are, on behalf of the panelists, we're going to be talking about the current and future impact of AI and ML on novel target discovery. My name is Puneet Batra. I built the machine learning for health team at the Broad Institute, and then was at Flagship Pioneering building preclinical discovery platforms using AI, and now I'm at a new organization called Ember AI, where I'm a partner, and I'm excited to be here. So we'll go through a couple of questions with the panelists. I'll introduce you before the first question, and then we'll open up the questions to the audience. You know, this is about AI, and everything is moving really fast, so throw at us whatever you've got. OK, so the first question is about doomers and zoomers. We're going to start light here. So in the AI world, in the anticipation of artificial general intelligence, an AI that can do any task at the level that a human can do it, or artificial superintelligence, where an AI can do tasks at levels extremely far beyond what humans are capable of, the field, of course, has fallen into a binary classifier. People are either classified as doomers, who believe that this is going to be a problem for the world, and we need to figure out how to get ahead of some of the negative consequences, or zoomers, who believe we'll be in an age of abundance. So, first question to Alex. Are you a doomer or a zoomer?
Well, first, thanks for having me here. I'm looking forward to the conversation. I personally actually lean, maybe I'm an optimistic, I lean to the zoomer side. What I find interesting is, of course, I'm at Roche, and we have a team both in San Francisco and a team in Basel. The team in San Francisco is more closely affiliated with AI and computational than Basel. The Basel site, though they talk pessimism, are absolutely much more in the zoomer camp. And the San Francisco site tends to be more on the doomer side. Now, maybe they know more than I do, but I tend to be on the zoomer side of things.
That's excellent. Sorry, I forgot to introduce you, so let me introduce you after you've answered the question. So we're very pleased to have Dr. Alex McCampbell. He's the VP and Global Head of Research of Neurosciences and Rare Diseases at Roche, as he said. He leads a team of over 100 scientists, coordinating research efforts from target identification through IND-enabling studies. While at Biogen, he led the preclinical work on tofersen for SOD1 ALS, and he holds a PhD in neuroscience from the University of Pennsylvania, and has an extensive publication record on topics such as ALS, SMA, and Alzheimer's disease. So what's the indication that you're looking for that'll tell you whether you're right about being a doomer or a zoomer?
Oh, wow. Well, we'll get into this, but the whole question for me around AI is, can it be more predictive? And meaning, can we increase our probability of success in the clinic? So the first time that we really come up with an idea that was accelerated and we got a clinical answer that was informed by AI, yeah, I'll be quite happy.
All right. OK, next panelist, Kristina. So Dr. Kristina Kitko, she's the head of AI investments at Eli Lilly Ventures. She targets early-stage innovation across Lilly's 5 core therapeutic areas. She comes from an operational background within biotech as well. She completed her PhD in pharmacology and electrical engineering from Vanderbilt, and postdoctoral training in brain and cognitive sciences across MIT and Harvard. So Kristina, are you a doomer or a zoomer?
Oh, I live in San Francisco, so I think zoomer with a side of doom.
That's right.
No, you drive around San Francisco in a Waymo, and it's hard not to be pretty impressed with some of the scale that we've seen come out of AI. I think the doomer side of it is almost a philosophical, these deep questions about sort of the changing nature of work and what that means sort of long term. But sitting in an investor's chair, I think we always have to kind of be optimists that, we're going to be more predictive. We are going to sort of positively change the nature of work. We are going to make progress on really hard challenges, and technology is just one way of sort of turning that crank.
Yeah, I like the optimism. OK, and our last panelist, Dr. Kaykas, who's the Chief Exploration Officer and Head of Neuroscience at insitro. He leads the integration of human genetics, functional genomics, and ML to machine learning to advance therapeutic pipelines, including major collaborations with BMS and Gilead. He previously served as a senior investigator at Novartis NIBR, where he directed discovery efforts resulting in multiple IND filings and clinical assets. He earned his PhD in molecular and cellular biology from the University of Wisconsin-Madison, and now he will also tell us whether he is a zoomer or a doomer.
I'm going to take the third option. Neither.
A little bit of both.
Yeah, a little bit of both. A little bit of. I definitely, I think a lot of the zoomer and doomer is made for social media, and I think there's, there's a lot of hype that goes along with machine learning, and I think that's, ultimately that can be kind of somewhat good in that it actually, gets us interested as humans, but I also think it, it brings, negative connotation, and I think, machine learning will really, kind of ultimately, help us as, in society, but I think, one has to be a bit cautious and careful and, kind of let it mature in the right way.
So maybe not so different from any other scientific technology that we've developed over the last set of centuries.
Yep.
Okay. Next question, moving on more specifically into the life sciences. So what's your prediction for what the impact of AI will be in the next, I'll give you 3 time frames, single year, 10 years, 100 years. So you have the opportunity to be as far forward-looking as you like, or as narrowly pessimistic as you like. Kristina, maybe because you see the biggest range of opportunities in this space, let's start with you.
I mean, not to be facetious, but I would love it if in the next year AI could actually sort like calendaring in our inboxes. The amount of time within a pharma that you spend sort of sorting calendars for big meetings. And, there are only so many ways that you can do that with AI right now, and it's not great. So, no, facetiously, I think we have to be realistic about sort of how fast we unleash these things on our hardest problems, our sort of most protected data sets, things like that. So I see it right now impacting a lot operationally from my seat. We're getting better at sort of handling volume faster and getting smarter from volume faster. 10 years from now, I think there's a lot of optimism at Lilly that, the way that we make molecules, that as we start to learn from our own data as a pharma, we can actually get smarter about both chemistry and biology, so both small molecules and large molecules, that we can actually start to understand the sort of underlying logic. We hope that that exists, first of all. We're optimists. We like to hope that that exists, but understand that logic. Yeah, 100 years from now, I don't know. I like to think that we'll all be on islands, sort of co-processing all day. So I'm a nerd. I grew up watching Star Trek. I like to think that maybe there's a world where, we really do progress as a society, but we'll see if that happens.
Okay. Aj, I wish I could have recorded your answer from 10 years ago before you started in insitro, but given where we are today, 1 10, 100-year prediction.
Yeah, I think, now and 1 year in the future, I think it's really productivity. A lot of the stuff we do is actually becoming a lot easier. If you want to send an email, you can do that almost automatically, or you can at least do it much faster. Things like regulatory reports or other things are, much easier, much faster. So a lot of those things, kind of that's right now. Everybody's playing around with agents, but it's still a little bit, kind of wonky. Like you don't trust the agents. So we're not there yet. But just general productivity, and then 10 years, I'd really like to think that we could do a better job of, say, stratifying disease. And start to, kind of, crack things like monogenic disease. I think there's a lot of opportunity, with the kind of data sets that Target is generating to really stratify patients and start to think about, kind of targeted therapeutics, across ALS. 100 years, boy, that's really hard to predict. I think, maybe we'll all be like cyborgs and society will all completely change because, things are moving so fast. But maybe on the disease side, that we'll have a chance to really make a dent in a lot of the diseases that are out there in 100 years.
Will we need labs in 100 years? Will it all be in silico?
Could be all in silico. I actually think it will be. Yep. I think you think about how things have changed in the last 100 years, and you think things are speeding up.
Yes.
So things are going to change drastically in the next 100 years.
Okay. All right. preclinical scientist.
Yeah. So I mean, first of all, the first part I have to agree with my colleagues on. Operations is where everything is at right now, and it does accelerate things. We joke with our computational scientists a bit about it. 9 months from now, are they going to need a new job, or are biologists going to need a new job? It's not clear, because you're coming together so quickly. Things you used to have to queue up to have them answer for you. You can now do a natural language interface and just say, hey, what happened here? What are the differentially expressed genes in this data set? It happens. It's so fast. And then you talk about writing reports, interacting with regulatory agencies, that's the barriers that are just coming down dramatically. 10 years from now, I do think everyone, us included, we're trying more and more, you call it lab in the loop, automation, so that there's an easier path to, you have an idea, get it done, even have the data analysis automated and just iterate so things can move faster. We often, at least we talk about what do you do and how do you do it. And I think right now what we're seeing is great acceleration in how you do things. 10 years from now, maybe you start to see what are you doing really start to change. I think the hardest part, and saying the obvious maybe, the AI does its best when you've got massive data sets that it also can learn from. You know what the consequences of that decision. So that's why coding becomes really easy. Yep. It's easy for the system to understand and realize where it's made a mistake. Now you're talking about projecting clinical outcomes, for example. That's a really long iteration loop. I don't care, it takes 2 years to run a clinical outcome trial. So 100 years, maybe the first longitudinal studies on longevity will start really reading out. I think you're going to have experimental wet data even in the future to drive this, would be my guess.
Okay. Aj, you've been building a company to do this for the last 10 years. So first specific question to you. Insitro was one of the earliest pioneers of AI in bio, one of the earliest that I could remember, right? You guys were in the early wave with recursion and in silico. Use high throughput screening, automation, principled causal data generation, everything we think are the major bottlenecks for AI learning properly the rules of biology. What can you share about how your view and your company's view has changed over the last decade? Have you been moving faster or slower than expected? And can you be concrete about something that you've done that you think is entirely due to AI in that span?
Yeah, these are great questions. So I'll start with the faster or slower. I actually think the kind of machine learning and the algorithms and things like LM have moved a little bit faster. That is moving really, really fast, and that's been really fun to watch and be involved in. I think the kind of lab in a loop, kind of things where you actually have, automation in the loop and you can do that really fast has been going a bit slower. It's really hard to do that. I think data generation, kind of at the scale that we all want, has gone slower. That's a big challenge. Those are things, kind of when insitro started that we kind of knew about, but I think, we definitely pitched, that we were going to be much faster in the kind of data generation and, and the time it would take us to get, kind of into the clinic. We're just going into the clinic now, and so, it's taken a long time. But, kind of that comes with, learning and building and, getting ready for the next cycle. So these things are, you have to build on. They don't happen, kind of overnight.
Okay. Specific AI development.
Specific AI
That's
development.
Right. I think often we talk in generalities, and here you are.
So there's definitely places where being at insitro, I've seen kind of machine learning really work well. When you have very large high dimensional data, kind of where we're good at seeing things in in 3D, maybe 4D with time. You get bigger than that, more dimensions. It's very hard for humans to comprehend that. So if you've got like large imaging data, and we've done a large project now in ophthalmology where we take in OCT images and built a foundational model there. It is amazing what machine learning can do there. I'd say the same on, we've generated a lot of images on iPS-derived motor neurons. And being able to kind of then, stratify based on, kind of genotype is something that, kind of computer vision and machine learning does really, really well at. So, those are.
Has that led to biomarkers, new target discovery?
Biomarkers, new target discovery, both of those are things that.
Specifically due to AI understanding how to read these images at scale.
That's right.
And doing a GWAS optimization.
And really looking at, the high dimensional aspect of the data, where we're just not good at that. If it's a simple thing, we can, humans are much better. But if it's a complex thing.
Yeah, that's good.
And if you've got large data.
Kristina, Anthropic just acquired Coefficient Bio for $400 million, which a lot of people that I know are very excited about. The Twitter sphere is now saying that they anticipate life sciences will be the second largest market for them after coding, which has been pretty impactful already. The CZI, Chan Zuckerberg Initiative, just invested $500 million into new data generation to crack AI and bio. You're sitting inside of Eli Lilly. What are the forces shaping your world, your choices of investments? When you see all of this acceleration, how do you decide what to prioritize and where you need to invest versus just wait back and see what happens? And I think of that as a model for any organization, including this one that's investing in AI. How do you make these decisions? How do you understand what's going on?
Yeah, it's a great question, and I think we're pretty privileged right now at Lilly to be in the position where we can try some things, and investment is one of those means that we have to sort of partner on things that in the past might have been potentially too early for the organization to directly adopt, but actually through investing in a company and sort of going along for the ride, it really allows us to see around the corner. And I think to your point on Coefficient Bio, I think every pharma are talking about that right now. Yeah. The world of sort of where are the frontier labs going? I think a lot of folks view that as an acqui-hire and a signal that indeed, Anthropic want to be a sort of enterprise service provider to pharma in a way that potentially hasn't existed in the past. And so I think that's kind of keeping us on our toes in terms of, a lot of pharma are building agentic labs. We're doing lab in the loop. It's actually quite interesting. Samuel Stanton spoke in San Francisco about 2 weeks back, and one of the things he highlighted was, do you know how hard it is to talk to lab equipment? Yeah. And it's this problem that every pharma have, right, and no one wants to solve. And I think you're sitting there in the audience and you're like, well, great, you were just acqui-hired into Anthropic. Is that something that we're going to see? Yeah. And so it's quite interesting that I think we're going to continue seeing this sort of interweaving with frontier labs and continued investment, I think, across the scope of everything from early discovery to even things that touch clinical and commercial.
Are you allowed to share one investment that Lilly has made in AI that's had an impact on how it sees the world, operates, gets products to market?
No, I can probably share 2 different ones. One, certainly our partnership with NVIDIA, where we decided, let's build a data center in Indianapolis and let's enable ourselves to build our own next generation of compute. One, probably more on the outcome side, it's hard at this point to actually share. We would all sit here and say, well, we don't have the data yet for AI improving a clinical trial directly. We're all kind of waiting on, be it chemistry, biologics design, clinical trial optimization. Everyone's kind of waiting on that. I can say that one thing that we've done is sort of internal work to look at AI process optimization. Really boring, but in manufacturing. We are manufacturing a lot of tirzepatide right now. And if you look at how we switch actually production lines, one of the things that we've done is worked with an external company on how do we actually optimize that switching. We've gotten something like 15% cost savings on that. It doesn't sound impressive, but
at your scale,
on a sort of, on the scale at which we're manufacturing, that's actually quite impressive. So a lot of the sort of logistical boring things are actually where we're seeing measurable organization improvements right now.
Thank you. Alex, what's it like from inside the lab? Preclinical research is where I would have said from my position, we're seeing the most promise and success with models like AlphaFold, et cetera, bolts that are able to co-generate molecules with proteins. What are you learning about applying AI internally? And is it autonomously in the lab? What are the challenges and opportunities in preclinical research? And same question to you for specificity. Can you point to anything specific that you've done where AI has qualitatively changed pace or discovery?
Yeah. So sort of alluded to earlier, yes, there absolutely are teams that are trying to do more and more lab-in-the-loop kind of approaches, absolutely. This, I would say, still remains at the experimental stage, really, of trying to see how does this work. Anytime you start doing automation, the more routine something is, the better, better off. So obviously it's going to be the screening facilities that are going to do more of that than, say, the research lab who's going to grow this iPSC cell today and this other cell tomorrow. And so that's harder to automate. But anything that is routine, absolutely. And then you have those fantastic learning cycles where, again, I think the advantage that a larger pharma has, you talk about the richness of data that exists, having all the PK data you've ever put, of any compound you ever put into an animal. Well, now you can start getting reasonably predictive models. It's all about how do you test less, right? That's what we're really aiming for. So that's sort of on the research side of things. Then the question is, can we get better at identifying what are you going to test against? And that's where collaborations like ours with recursion kind of come in, right? Are we actually able to get interesting new targets out of that? AI has a common problem of, which I remember back when I was a grad student, microarrays were coming onto the scene, right? And microarrays are great. You go profile your tissue, and then everyone says, well, do you have the thing that everybody else knows that changed? And you hope that you see that. And then you look for the thing that looks right next to it because that's related but still has plausible biology. If no one's ever heard of that gene before, you don't care about it. And I think AI kind of has the same problem when we start talking about target discovery. If it's too far away, you're by yourself, essentially. You have nothing else to anchor it to, so you're always looking for that Goldilocks target that's similar to something you've seen before, but not the thing that everybody else knows that they're studying. Yeah. And I think that's what we're all looking at right now, are those adjacencies to see, can we really bring something like that forward and bring that into the clinic as an example to say, hey, look, this was really discovered by AI.
And so you haven't found those yet, but you're optimistic?
We don't talk about a research pipeline. No,
I'm not asking for specific indications.
No, we absolutely have things like that. Absolutely, and but this is where the proof is in the pudding. Ultimately, it's going to take time, right? It can look promising for sure, but until you actually get it into the clinic and you can actually show that it moves things, you don't know. And the first few may fail. Our drug industry is a game of probabilities. I think probably everybody in this audience knows roughly 5% of things that enter phase 1 make it to the market, right? If AI can get us from 5% up to 10%, this is a huge success. This is absolutely an unqualified success. But that still means that 9 times out of 10 have failed.
Yep.
So it's going to take a little while.
Aj, I saw you nodding along a lot.
Yeah,
no, target discovery?
I think we're still early in this, in AI, picking or helping us pick targets, helping us actually, make therapeutics. And so I think over the next, 5 to 10 years, we're going to actually have enough shots on goal to where we're going to see, are we doing better? Are we actually doing better? Is in sitro and silico recursion, now pharma companies that are actually here, are the targets they've picked, are the drugs they're making, do they have a higher probability of success than, kind of what we've done in the past? So I don't think we know yet. I think we need to kind of, we've got this, we've got this thesis. This is going to, kind of help improve the probability of success, but we don't know yet, and so we're going to have to see what happens.
And where do you think the breakthrough will come from? Is it going to be in the foundation models like AlphaGenome? Is it going to be in the multimodal models like EvoScale? Is it something we haven't seen before? Is it a new, data collection assay like 10x is producing that'll do this? What do you think is the gap? Because 5 to 10% is incredible, like the number of lives that would affect is amazing, but this is not what we hear the AI prophets telling us. This isn't going to be a 2x. This is going to be a 100x in 10 years.
I mean, I think there's a lot of problems wrapped up in kind of what you said. So I think there's making better therapeutics. I think that's really improving. We're getting much faster at that, and that's because of things like kind of AlphaFold and all of the ability to actually do machine learning on on structure. So that's getting much faster and better. I think target discovery is still really hard. Kind of, and we all love genetics, but there's not enough data out there. Kind of, there's not enough way to kind of validate those targets. Like I think target validation. There's targets out there. There's genetic targets, but how do you actually validate those at scale? and pick the best one. And that's where I think there's still a lot of challenge, kind of in what we're doing. And I don't think we're going to have breakthroughs there, kind of really quickly. There's a lot of people talking about virtual cells, and that's going to really help us. And that's a really good, that gets us motivated, but, cells are complicated. And just by doing perturb-seq on a cell, and looking at the transcript, you're not going to get the full essence of a cell. And so I think there's a lot more data and collection that are need to go into that to be able to build foundational models that are helpful for us. So I think it's really in protein design and small molecule design that actually I think we're going to make headway now.
So let's use that to lens directly into ALS now. Kristina, something you said before has really stuck with me, that we cannot afford trial failures because that could have a company starting to look for greener pastures elsewhere where there's easier money. So we want to be able to validate the targets that the ALS community is going after. How do we do that? How does AI help us with that? We know it's a hard problem, but what do we need to get more successful at that?
I get to start, great. Yeah, I'm a neuroscientist by training, and it's one of the things that as you're doing a PhD, as you're doing a postdoc, you really struggle with is the sort of enormous challenge of all that we feel like we know and all that we don't really know. And, if you look at sort of, new molecular entities in the neuroscience space broadly, it's just hard, right? It is really hard, and the conviction that you need to have around a target and the timelines that it takes and the challenges with running trials. And so, we try to be really thoughtful and really careful about, how we approach this. And the question was?
Oh yeah, what's it going to take to improve target validation in ALS?
Yeah, I don't know, and I don't know that there's one single simple answer to that. I don't think that we can sit here and, faithfully say, yeah, AI is gonna, for ALS, sort that all. It can help us with bodies of evidence. Labs in the loop can help us collect more data, get more target conviction. We can better understand patient populations, and this is where I think organizations like Target ALS are really doing incredible work to continue to fund, new target discovery and understanding patient cohorts, and really building trust with the community, because I think all of us on the stage here believe that data are key.
Yeah.
And so actually continuing to collect that data, not just inside of a pharma, but fundamental scientific research is, I think, ultimately going to be the foundation of what it's going to take to sort of crack on and move forward.
Yeah, and maybe just to put a slightly more, I don't think you're being negative, because I think it's a really hard problem.
Yeah.
But maybe I'll phrase it slightly more positive, because what we've seen at this conference are people proposing different ways to predict outcomes, different biomarkers that might be useful, deeper and deeper phenotyping and collection of clinical courses. This all becomes a feedback loop, right? And this is what we will need. None of us are going to argue that we'll have the magic answer next year, but we're setting this organization is setting the stage so that you'll be able to start making intelligent guesses, maybe AI intelligent guesses, and that will then feed back. And they might at first be wrong. Then it'll get better and better as you start learning which of these new biomarkers that are emerging are actually useful, which of these patient stratification strategies are actually useful, which of these are these perturb-seq models, are they actually starting to predict outcomes in a meaningful manner? I think the challenge with AI is it doesn't have that experiment, it doesn't have the end goal yet. As
we can't close the feedback loop.
You can't close the feedback loop, but that'll start being closed over the next few years.
Aj?
Yeah, I think collection of massive amounts of human data, whether it be clinical data on patients or on cells. I think we've the animal models for ALS are pretty challenging. Kind of we all know that, and they're good. They're good for certain things. I'm not like we want to target engagement, but I think for efficacy, there's not a lot of success there. So I think the more that we can focus on kind of human data sets and try to bridge the gap from the kind of mechanistic biology that we can learn from in vitro systems or even, kind of organoid systems and bridge that to the human data, I think that's where, machine learning is actually going to really help there to kind of, put those data sets together and really help us make better decisions on, what's the right target and what's the experiment to do that will actually, help us validate that this is a good target, that this is something we should move forward on.
Can I ask you a question? Have you used AI to guide investment decisions yet?
Oh.
No, the joke is at Lilly, we are the first people that will be replaced. So, we don't use it to guide decisions, but it is actually pretty good at laying out rationale and thought process. And, I think all of us on our team kind of co-work with AI day in and day out. And so it gets pretty good about understanding, like, hey, Kristina's going into an investor call. What's she going to ask the person? Like, it's actually pretty good about, it'll tell me what I'm thinking before I say it. So that's quite frightening. But no, I will say we're not pessimists. I think what we see it as, this is all a partnership. We are here to be good partners to the ecosystem, and we know it's hard, but this is why we do it.
It's pretty good for personal investing. It outperforms me, for sure.
So we've still got a couple more questions we can go through, but if there's questions from the audience, happy to take them now as the discussion period has started. You can just line up to the mics.
I have a couple of questions. So I lead a clinical biomarker and precision medicine team, and my team is asked every day, give me a biomarker that will predict trial outcome, right? And I want that biomarker to move within the first month in a phase 1b study with 12 patients, right? So, an impossible task. And I was wondering, one, it seems to me, we talk a lot about absence of data, we need more data, but really thinking about, and I'm at Sanofi, thinking about, there's enormous amount of data that Sanofi has collected over the years. Granted, right, and I guess when I think about this, I think big pharma is in the best position to generate the data that's needed because we have these, super well characterized patient populations, right? We have the clinical data. What we don't have is a lot of sort of, molecular data. And that's something that I know a lot of companies are investing in, including Lilly and us. We're generating now proteomic data, transcriptomic data, genomic data. But I'm wondering, how do you think about, at least from my perspective, it seems that at the end of the day, what it's going to take is for big pharma to unite and decide to actually share the data. Otherwise, I just don't see how we can kind of work in these, vacuums. And there's some interesting examples from Europe in particular. For example, on the Alzheimer's side, there's this big initiative, AD Riddle, a variety of initiatives, where essentially the academia, right, has joined hands with big pharma and is trying to bring together these very large data sets. So just wondering about your thoughts in terms of how can big pharma partner? Is that something that you're also thinking about? Is that something that will need to be maybe stimulated, by, I don't know, the government, although our government will.
So maybe just summarizing what's it going to take to acquire these data sets, and is pharma willing to do it?
And to share, right?
Because and to share that.
Yeah.
Great
and
question.
I have just a quick second question. I get approached by a variety of companies with these foundational models where they are wanting to partner, around trial outcome prediction. Can you give me maybe an example of two that you think are worth partnering with?
Great question. So 2 questions. Anybody want to take the first question about pharma data consortia? And then I think the second question probably is best for you, Kristina?
So farmer data consortia do exist, and Roche participates in a number of different programs in which we and others are paying to generate data. Back when I was at Biogen, they created MS Paths, and I know other organizations are involved with that. So these do exist. I think that the data, could we do more? Probably, arguably, yes. You always have to negotiate all the complex legal frameworks, and this is really pretty competitive, and how do we enable everybody? But those are all things we can sort through. I actually think to some extent, though, I'm going to say it's a quality of data, not critiquing the technical quality, but it's the lack of intervention and association. So all you're left with is correlative data.
Yeah,
not causal. And we don't know what of that correlative data is important or not. And I think until we solve that problem, we're going to be a little bit we have a lot of data, but not much information.
Aggregating more confounded data isn't necessarily going to tease out what's going on.
Yeah, I would just add on, we're also members of a number of consortia, and certainly we see, if you can get through the negotiation phase of, legally protecting everyone, maximizing upside, all of that, they can be incredibly beneficial. I wanted to address the clinical side also, because you have to remember that, at least, the way we see it, you have to also obtain patient permission for use of clinical data. And so oftentimes people say, well, pharma are sitting on all of this clinical data. Actually, the patient owns that data.
It's not reconsent.
We would have to then go back and reconsent in all of these trials. And so, I do think going forward, there will be sort of thought put into that by pharma about how we actually obtain consent for use of data in certain ways. But we always try to be very careful and say patient owns the data, and we're not retroactively going back to touch that.
And then very quickly, are there any two of your investments you would love to promote right now for predicting probability of technical success in clinical trial outcomes?
Well, let's take that one offline.
Gotcha. She's available afterwards.
Complex.
Okay, let's take a question over here. Thank you.
Sure, thanks. I really appreciate this discussion. I wanted to just get at a question about the difference between precision and accuracy in all of these machine learning methods, because there seems to be a real focus on precision of the predictions it's making without a real understanding of what it's learning and why it's making those predictions. And so I think, this thing about how it takes high-dimensional nonlinear data and can see that, right, in ways that maybe we can't. That data analysis part is what it's really great at. But I wish there were more focus on letting it do the data analysis so we can do the data interpretation. Visible by design methods that are as powerful as some of these black box methods, and really let us understand the underlying biology rather than just having a list of predictions. I'd love to hear what you think about that.
Anybody excited about those methods?
I can start. I mean, I think, we tend to not like the black box models at insitro. We like to, kind of be able to understand what the models are doing. That is something, that being a biologist, I want to understand, what's behind all of this. And so, the way that we've been thinking about this is really not, is kind of, there's a co-scientist or, co-agents, and you need a biologist there to kind of interpret the data in the end. And so it doesn't, kind of do better than we're doing when we, understand the data and understand, how the algorithms work.
So I definitely don't disagree with that. But I will say that if you think of something like AlphaFold and how much value that has brought to being able to design new proteins, et cetera, it has not elucidated a fundamental new insight into protein folding, right? There's not a new explanatory power that's come out of it.
Yeah.
But it's extremely powerful as a tool. So I will take that.
It is, but it's incredibly powerful because they built the biology in. They started with what we know about how domains fold and then used that to be the basis of the predictions that they were making. And this is what I want for other kinds of predictive models, right?
It started with a bunch of structures, a whole bunch of structures that were known,
Okay.
right?
Yeah. But what it's predicting is how those domains fit together into those structures, right?
I mean, in some of the biology that we do, we use, kind of convolutional neural nets to kind of be able to classify, images. You can do ablation studies and figure out, what's the algorithm seeing. I mean, these are things that really help you interpret. Or you build GANs, and you can actually, then apply an LLM on top of that and actually understand, okay, this is what the algorithm is actually seeing. This is why it's classifying. Even if it's a self-supervised, kind of, learn. And so that's been really, pretty exciting for the biologist, because we want to know, like, why are these images being classified differently? It's, if you can't tell what it is, it's like some, high-dimensional thing. You're like, I don't know what that means. So you need to be able to kind of understand what, what it means.
I would like to learn too, just for what it's worth.
Thank you.
Thanks.
Please.
Hi, thanks so much. So clearly all of you up here are embracing AI to different extents and in different ways. We at Target ALS are really excited about the potential of AI reducing those cycle times from getting, identifying a therapeutic target to getting to a therapeutic in the clinic. I'm wondering what advice you would give to people in the audience here, or people who see the live stream who are not yet taking up AI and feel very uncomfortable with its ability to help them do their jobs. What would you recommend that they try? Because there is an element of knowing how to use AI, if you will. What advice would you give those people to encourage them to try to incorporate it more in their workspace?
Great question.
Yeah, I can start. I mean, I would say, make sure your data is good. Make sure the quality of your data is good. I think that's the first thing to start with, and then don't worry about, kind of building your own algorithms. Those are changing so quickly. Kind of learn about, what's a good algorithm now, try it. As long as the data you have is of high quality, and you've actually, I would say, like, everybody here should be thinking about, how to capture all the metadata, how to make sure that, kind of everything is done in a way that you could apply machine learning to. And I think right now, a lot of the challenges with what I see in machine learning is really, it's a kind of a data quality problem. It's enough data, it's the data quality, and so focusing on making good data.
I think the other thing I might add, I certainly agree with that. The other thing I might add to it is that not to be afraid. Often we say, AI hallucinates, AI can make mistakes. That's absolutely true. As you start using the tools, you figure out how to interact with them in a way that it can reduce that dramatically, right? And then you start realizing the true value of it. There is, what you'll find is that people that get good at this elevate their overall ability to get more done. It's an enabler. It's a way for you to expand your reach of what you can interrogate. And if you can figure out how to get AI to be drafting a paper while you're designing the next experiment, more power to you, really.
It's pretty fun to start to, like, do vibe coding. I mean, everybody can do that. Yeah. So, I mean, that is something, and you can learn about it. And some of the people that are actually doing the best coding are not people that were trained as, kind of computer scientists or engineers now.
That's right.
So it's a different world.
I don't know. So I will give the counter to that, which is I came out of MIT, and when I was at MIT, AI was math. I tend to fall back on, machine learning in the old school days was sort of statistics. It was math. And so I actually got into machine learning because I was a neuroscientist, and the size of the data sets we had were such that practically the only way to sort of stay on top of my day job was to do machine learning. And so for me, I actually started by keeping it very bounded, that it's doing a very particular task in a very particular way, just at scale. And that kind of gave me the confidence to sort of build up to, like, okay, now, Claude can help me pull papers and things like that.
Great. Okay, so we're out of time. Lightning round. Quick answer and a reason why. Claude, ChatGPT, or Gemini? Aj.
I've started to use Claude a lot. I like Claude for Life Sciences is good. I like the integration with kind of Claude Code. We have a desktop version. It's actually integrated in now with a lot of Slack and my email and my G Drive. So it's integrated. You can pull things from external sources. So,
Kristina.
Yeah, we are a Claude enterprise, and so anytime that you can actually sort of have Claude help you do your day job, I I have to say it's been pretty wonderful.
Alex?
I'm a faithful and dedicated polygamist, all three.
And they talk to each other and do stuff on the side.
They each have their strengths. You know, if you want to do coding, Claude is fantastic. We use Gemini for a lot of things. That Notebook LM tool they built is fantastic for interrogating things. And then ChatGPT, AI tools have their own real good use. It tends to be a little more rigorous on certain questions.
All right. Thank you, panelists. And thanks, everyone.
Thank you. Yeah.